I'd like to welcome Dr. Berry up now, who's co-director of the MGH ALS Clinic. He's associate director of the MGH Neurological Clinical Research Institute and director of the Partners Neurodegenerative Clinical Research Fellowship. He's an ALS clinician and clinical researcher, and he has designed and led ALS trials, conducts research aimed at biomarkers to help us understand some of the disease biology and to act as outcome measures. He also serves on the board of the Northeast ALS Consortium, and it's also been a privilege to work with James all these years. Jim, come on up. He's going to talk to us a little bit about clinical trials and how we design them.
Yes. Yeah. So I'm going to talk about some of the principles we use to design clinical trials rather than specific ongoing trials. Oh, look at that. And so just to sort of set the stage, I would say that there are a few ways that we go about drug development in ALS, or drug development in general. And the first one is sort of what we would call targeted development. That is, we sort of focus on a disease cause. We do sort of some of the elegant work that Jonathan has talked about, sort of looking for specific molecular targets, and then we build a program around that. And so one, example that comes to mind is, a mechanism that seems to be important in ALS is oxidative damage, and that leads to a compound like edaravone. And this isn't a perfect example because it wasn't designed specifically for that, but that leads through that biology to an idea of something like edaravone, which you then put into preclinical and clinical testing. The other way that we develop drugs in ALS, and again in other diseases as well, is just through clinical observation. And a good example of this is Nuedexta, which was in trials in ALS to see if it slowed disease course, and during the course of those trials, it was observed that it had an effect on pseudobulbar affect, and that led to a drug development program to look at its effect on pseudobulbar affect. It's a little harder to develop whole programs around, sort of a continuous program around clinical observation, so those tend to be sort of a little more catch as catch can, but really an important way to find ways forward, and there are many, many, many examples throughout medicine of how this can be a successful drug development strategy. So both are viable strategies. Once we have a candidate, then we design a series of trials in humans to sort of see what the effect and safety of that molecule will be. And when we talk about trials, the trial is essentially a tool to get from having some molecule to having a treatment for a disease. And so trials are not in and of themselves the sort of end game. And part of the reason we have different designs of trials is that every drug development program has slightly different needs. But I'll try to talk about some of the unifying principles. So kind of an overview of novel drug development is that we have a molecule and we want to get that to the people who need it, but there are a lot of steps along the way. And so the first step is really basic science. It's drug structure and production and chemistry, and this is after we know what we're targeting, by the way. So much of what Jonathan talked about happens even before this step. So we need the target, then we develop the drug, and this is heavy on the kind of chemistry and molecule design. And then we can go to maybe cellular models and then animal studies, and we look at safety, we look at dosages, we look at effect if we have the right model. In this case, we have an SOD1 mouse model that's used most commonly, but we have a growing number of ALS models that we can test things on preclinically. And when the time is right and we have enough of this safety data and enough evidence to move forward, then we go to the FDA for an investigational new drug approval, which allows us to then head into patients. And when we get to patients, we have kind of a very sort of semi-regimented way of going through the paces so that we both move a drug forward, learn what we need to learn, and mitigate any potential safety or toxicology problems along the way. And so the first step is phase 1 trials, and I'll talk about a typical phase 1 trial design in just a moment. But this is the first time we give this to people, and the main thing we're trying to get out of this is it safe? Is it at first blush, is it safe? Do people have a terrible reaction to it? We can try to get more than that out of it. We can talk about where the drug goes, et cetera. But that's the main goal. If we pass that step, then we move into phase 2 trials. And phase 2 trials are actually probably the most complex step here, because we're trying to move from, at first blush, this drug is safe, to through sort of understanding where the drug goes, what doses are right, can we follow some marker to see whether we're engaging our target? Are we changing disease biology? So there are a lot of questions, and in fact, many drug development programs include more than one phase 2 trial. There may be very specifically designed phase 2 trials. So we're answering a specific question about where the drug goes, or a specific question about dosing, and then that's followed by a specific question about what the best outcome measures to look at are. So we might have a series of small-ish trials that prepare us for the final step, which is the phase 3 trial, which is what we call an efficacy trial. And just so that we're not lost in words, efficacy is kind of like effectiveness. There are subtle changes, but it's sort of under close observation, what is the effectiveness. So think of effectiveness and efficacy as very, very similar. Once we get through a phase 3 trial, if we have a positive result, that is, we've changed the course of the disease or had an effect on the symptom, if that's what we're targeting, then we send that data to the FDA to review for final approval. And all of the data gets sent. It's not sort of, well, it worked, and here, believe us. The data has to be set up in a very specific way so that they can analyze and reanalyze that data, and they typically require 2 trials that show the same thing. And then we can get to people, to sort of broader population outside of trials. Now, we can save time if we can repurpose drugs. That is, there may be drugs that are already on the market that have gone through the basic science and all the toxicology studies. We might want to repeat pieces of that, but we can very quickly move through that. We can sometimes phase 1 trials have been done, and it's satisfactory to move straight to phase 2 trials. So we've saved years and years of time by finding a drug that does what we want it to do, but we're moving it to a new disease indication. And there are many, many examples of ongoing trials like this, retigabine, maxillatine, Actemra. So there are a host of sort of repurposing programs with the promise of targeting a pathway that we think is important, but doing that without all the cost and expense of making a new molecule. They still have to move from phase 2 trials into phase 3 trials, and then the final part of this pathway is the same. So just to give you a sense of what do each of these phases look like? A phase 1 trial design is there are lots and lots of variations on this, but there is a canonical kind of, this is what we think of when we say phase 1 trial design. And it goes in groups or cohorts. And so the first cohort, remember, these are the first people to ever be exposed to this drug. The first cohort gets a very low dose of that study drug. So when we do our studies in animals, if our dosage is 10, and that dosage causes just some slight side effects in the animals, we would start at 1 or maybe 0.1 in people, a very, very low dosage. And this is often a single dose. So really, really starting in a conservative way. But again, this is the first time people are exposed to this compound. We then, after we treat typically 3 people, then we move to the second cohort of 3 people, and we might go to a middle dosage or up 1 dosage. And how we do that depends on very much on the drug. And we might go to a third cohort at high dosage. Now, sometimes there are 5 cohorts, and sometimes there are only 2. Sometimes you have 6 people in a cohort, and sometimes only 3. So there are variation here. But really what we're looking at is safety and tolerability. We may also be looking at the half-life of the drug. How long is it around? And finally, sometimes we look at whether it gets into the central nervous system by collecting spinal fluid. So the main thing we're looking at here is safety, though. Phase 2 designs, as I said, are probably the most complex. And so it's actually very difficult for me to put up one slide and say this is the sort of canonical phase 2 trial. But we'll walk through this one and say maybe this is as close to kind of standard as we might say. It's a larger group of people. It's not a huge group of people. And that can vary dramatically. A large phase 2 may be 100 people. A small phase 2 may be 10 people. There is often a randomization, and I'll come back to this idea of randomization. but there is often a randomization so that people are placed into different arms or different treatment groups. Many times there are multiple dosages, so there may be a high, a medium, a low dose arm, and there may or may not be a placebo arm. Many times there is a placebo arm, and that I'll talk again about placebo, but that helps us understand both efficacy and, to some extent, safety. You can have open label designs where there is no placebo. Sometimes there's a single arm phase 2 study, so again, there are variations on this. Now, the other thing that's complex about this is different phase 2 designs might look at different outcomes. But here are some of the things that we're often looking at. And many times we're looking at many of them at once, though we do have to declare one our primary endpoint. So safety and tolerability. Now we're giving multiple dosages. So we may be primarily seeing if this is safe over a longer period of time. Drug levels. Maybe we're looking at how the drug, what the level of the drug is in the blood. Or maybe we're looking at the level of the drug in the spinal fluid to make sure that it gets to the brain or spinal cord. This is a simple concept, but there are actually a fair number of drugs that have failed in ALS drug development because they just didn't get to the brain or spinal cord the way that we thought they should. We may be looking at markers of biological effect of the drug, and we sometimes call this target engagement. That's sort of the term of art. Maybe this drug is supposed to interact with the receptor, and we can measure the levels of that receptor or something downstream of that receptor, and we can just say, is the drug actually getting to what it's supposed to get to? Is it supposed to reduce a kind of cell in the blood? And we measure those cells in the blood, and it's reducing that kind of cell. Okay, it's active, at least. Doesn't mean it's working, it just means it's active in the way that we thought it would be. We also, I think, increasingly have markers of biological change in the disease. One of these is something that Jonathan talked about, which is these dipeptide repeats in people with C9 ALS. There are others, and I'll show others, but neurofilament, we now have some PET scans. So we are building cases for a variety of biological biomarkers that reflect some aspect of ALS. And that's different than a few years ago, and I think it will be a big improvement. It will allow us to have much better clinical development programs. And then finally, efficacy outcomes. So phase 2 studies do not answer the question, does this drug work or not? But we can begin to build in outcome measures like the ALS functional rating scale, survival, strength measures, breathing measures, to prepare ourselves for a phase 3 trial. I think I have to tell you, for any of us who design a phase 2 trial, there's a little part of us that hopes we've got a home run and we're going to see some effect in phase 2. And I would be lying to you if I said I didn't think that every time I was involved in designing a trial. But the reality is, that's just not how it works. You hope to get some hint of a signal, you hope to understand which outcome measures are going to work best for your program, but what you really need is a phase 3 trial. And these are some of the simplest designs in many, many respects. You get a lot of people, you randomize them, you test the study drug against placebo. That's the gold standard. There are ways to do this without placebo. They're complex, and they come under a lot of scrutiny because they don't, and I'll talk about that in a minute, but they don't answer the question quite as reliably. And then we have an efficacy measure. Is it working? Are people surviving longer? Is their function better? Are they stronger? Are they breathing better? And that's the whole thing. We have one dose by then, generally, not every time. Generally, we have one dose. We test against placebo. One of the questions that comes up sort of anytime we talk about trials, and I think appropriately so, is why do we need these placebo groups? And I think tied to that is what is placebo and how did we come to use placebo to begin with? So a placebo is an inactive substance that looks exactly like the active drug and tastes and smells, and those are important things. Actually, taste can be a really important thing. Designing placebos is actually no small feat. In fact, it's a substantial line item in the budget of a trial many times to develop a placebo alongside the active treatment. So the placebo refers to this substance that's just like your drug in all respects except that it's inactive. We also talk about a concept called the placebo effect. And the placebo effect is this. Our brains are wired deep, deep down. In our sort of reptilian, the dinosaur part of our brain, it's wired to look for cause and effect. And the thinking about that goes like this. Oh, my friend Bill just ate those red berries, and now Bill is dead. I shouldn't eat the red berries. That is a very important part of who we are as people, and we cannot ask. There's no way to ask people to put that aside. It's a reality, and we have to deal with that reality. The problem with it is that we sometimes do this inaccurately. So sometimes there is an inert substance that somebody takes and then reads into that an outcome which is actually not in real life attributable to that inert substance. And if that inert substance is truly inert, that's fine. In fact, they've done trials where they say to people, here, these are placebos. They have no biological effect, but you might feel better if you take them. And people feel better. And I'm sure I'd be the same way. This is how we're built. But if we say, we think this may be a treatment for ALS, we want you to take this, you may feel better. It can be very, very powerful, and people can feel better, at least for some period of time, without a true biological effect of that substance. And what we're looking for is a biological effect of the substance. We're not above being happy about a placebo effect, but we're looking for a biological effect. And so we use the placebo so that we can create an even ground. We're giving everybody some substance and saying, this might be an active substance, and that active substance might have an effect on ALS. And that sets the ground, and now we have a comparison group. So in a trial, people who are receiving the placebo are this comparison group. And I should say this has to be done in a blinded way. So if I'm the investigator, I can't know who's getting placebo and who's getting the study drug, nor can the person who's actually receiving it. So let's imagine a trial where we're looking at ALS functional rating scale. This is a 48-point scale, and we can see sort of some decline over time in people on the scale. And let's say we did a trial that was just active drug, and we saw a decline in people in the trial like this. It's hard to know what to make of that. Now I didn't put numbers on this, and I didn't put them there on purpose. That we have some concept that on average a decline of ALS-FRS is 1 point per month, but the truth is that actually varies a lot if you look trial by trial, because we get different populations in each trial. So it's a fairly uninformative result. If that line were going up, it may not be uninformative. That's what we all hope for, but the reality is that we've seen a lot of trials like this. So this is called factual. We gave people some substance. This is the factual result. We need to know what we call the counterfactual, which is, what if they didn't take that drug? What would happen there? And that's what the placebo group shows us. People who didn't take that drug. Everything else was the same, they just didn't take that drug. And so what if the placebo group looked like that? We'd say, well, I don't think we have a drug here that we should take forward. But if the placebo group looked like that, maybe we've got an effect. And we have all kinds of fancy statistics to say, are those actually the same line or are they different lines? Is the trajectory actually different? What's the chance that trajectory is actually different? That's what all the statistics are about. That's what p-value tells you. But this is really what we're looking for. What is the factual if someone takes the drug, and what's the counterfactual, that is, if they don't take the drug? And one point to make here is that when you walk through this exercise, you realize that the information from people taking placebo is just as important to the success of a trial as the people taking the study drug. For this kind of trial, we cannot do it without the placebo. This is why this becomes a part of the standard trial design in many, many, many cases. Now, I also happen to believe that we need to look for ways to do this without placebo, and I'll come to some of those ideas later, but I will also say that this is the strongest evidence. Methodologically, this is the best way to get this job done. There's something else that I've talked about, and I've just sort of thrown it out there, which is randomization. And randomization is a process of using chance to allocate people to the placebo group or the treatment group or the high-dose group or the low-dose group. And so, why randomize? Well, that's an important process. First of all, it's important because otherwise, how would we decide? I mean, it's very infrequent that people come in and say, I really want to be in the placebo group of a trial, please. Mostly people, given a choice, would choose to be in the active group. And so this is sort of if you leave it to chance, that's a fair way of allocating. Beyond that, what chance does is allows us to balance characteristics of the people who are in the two groups. So age, we hope, will balance between the two groups, and disease duration, and whether it started in the bulbar region, the facial region, or in the arms or legs. We hope that those things will balance out. And they do, basically. I mean, it's essentially flipping a coin. You basically get 50% heads and 50% tails. And that's what randomization does for us. The problem with randomization is that just because we randomize doesn't mean that we get equal groups. So just because you're flipping a coin doesn't mean that you get 50% every time. And it actually turns out that the more times you flip a coin, the closer you get to 50-50 heads and tails. So in very large trials, randomization works quite well. But in smaller trials, there can be a problem. We can have meaningful differences in baseline characteristics between groups. So let's imagine a trial where we randomize, but it turns out that the placebo group average age is 70 and the treatment group average age is 50. That's just how it worked out. And so we have a second tool that we use to try to, and this sort of demonstrates that. You know, if you randomize these people, okay, if some people are purple and some are red, you know, it's not that far off, but are those two groups really equal if red or purple is an important characteristic? Actually, probably not. So if we really want red to be randomized and to be equal, and we really want purple to be equal, we have to add a step to our randomization. And that is something called stratification. So stratification helps us reduce the differences between groups due to chance. And we can ensure that a few very important variables are better balanced between the groups. And rather than go through explaining it, I'll say that we sort of create two different randomization schemes and fit people into each one. So for example, red gets its own randomization scheme, and purple gets its own randomization scheme, and it kind of looks like this. Okay, and if there were more reds, they would go into the red groups. If there were more purples, they'd go into the purple group. And that allows us to do a better job on a few characteristics. So that's sometimes something that you hear about. A word about heterogeneity and subgroups of ALS as well. So in order to sort of just demonstrate this idea, because we throw it around a lot, but just in order to demonstrate it, we're going to do a little experiment. The experiment is going to be how quickly people, all of you, can find the red dot. Here we go. Pretty fast, right? Let's try it again. That one takes longer. Okay? This is a demonstration of heterogeneity and its effect on an experiment. So heterogeneity just means diversity in a group or differences in a group. And ALS is by its nature heterogeneous. We can think about clinical parameters. So it can begin in the face, the arms, or the legs. It can spread to multiple regions or entrench in one region or a couple regions. It can progress rapidly or slowly. It can be accompanied by fatigue, weight loss, cognitive change, pseudobulbar affect, or none of those. So there's a lot of heterogeneity that's built into ALS, and we can't change that. That's how it is. So we have to figure out what the best way to deal with it is. One way to deal with it is to do bigger and bigger trials, and that helps us statistically deal with it. Another way to deal with it is to do something that we call subgrouping. And subgrouping would allow us to do this. And sort of maybe we do a trial that only includes the light blue so that we can see that red dot more easily. Okay? The difficult thing about this is that, number one, there are fewer blue dots than there are all dots. And so it takes longer to enroll. But also, what if you're not a blue dot? And I think that's something that we've been talking about a lot recently, because the trend in our clinical trial design is to try to reduce heterogeneity, because we think this gives us a better chance of finding effectiveness in drugs. So let's talk for a moment about subgrouping and how it affects trials. So if we do this appropriately and choose appropriate subgroups, not random subgroups, but subgroups that are meaningful to the effect of a drug, we can reduce trial size. We can increase what we call statistical power. That is, what's our chance of seeing an effect? So we increase the likelihood of success, and we also, another way of saying that is reduce risk. That brings, everybody wants to reduce risk, whether it's a foundation that's funding a trial or industry that's funding a trial. If we can reduce the risk of that trial and say, your chances are actually higher of finding a successful drug if you do it this way, that brings more people to the table, which means we may be able to do more trials, and each trial has a higher likelihood of success. And so I think we should look at this as a way of increasing our hope that we tie to those trials. It may mean that there are 6 trials going on and a given person only qualifies for 1. Sometimes there may only be 4 with 2 more on the way. The 4 that are open, somebody may not qualify for. But there will be one on the horizon if we do this right that an interested person will qualify for. And so I think there are a lot of reasons that in the moment it's frustrating to have subgroup trials. But in the long run, it may be, if we do this right, again, if we think about this carefully, it's really a reason to be more and more hopeful about our trials. So I want to talk just briefly about how we use biomarkers in trials as well. This is just a couple of potential biomarkers, but there are a number of others. NFL is neurofilament light. So neurofilaments they're looking like a biomarker. They're elevated in people with ALS. They're not specific to ALS. They seem to be relatively stable if we look over time. There's a lot more work to be done here, but the higher levels of neurofilaments, heavy, light, actually either one, correlate with faster ALS progression. And so we may be able to incorporate this into trial design. There is also now early evidence about a PET scan that shows that we can image inflammation in people in the motor cortex of people with ALS, particularly those with upper motor neuron signs. And we may use this as a quantifiable biomarker in clinical trials. But how we use biomarkers in clinical trials is also complex. So we might enroll people with ALS who have certain biomarker levels at the time of enrollment, and there are trials that are doing this. So maybe there's a biologically relevant subgroup, people who have more inflammation, if you're using sort of an anti-inflammatory. And that way you get people in who are more likely to benefit. The very clear example of this is targeted gene therapy for SOD1, which is with us now. I mean, there's an ASO for SOD1 or C9. You want to target the population that has the gene mutation, but I think there are other ways that we can use this as well. We also might see if we can alter disease biology using these biomarkers. Maybe we can see those dipeptide repeats come down, and that sort of uses biomarkers as an outcome measure. Say we're changing the biology of the disease. Or maybe we can use a biomarker to predict how fast the disease is going and then see if we can change how fast that disease goes in that group. So maybe we can get people who are all predicted to be rapid progressors and see if we can turn that whole group into slow progressors. We're not probably quite there yet, but there are some disease prediction models that are really becoming more and more mature, that are helping us using complex algorithms sort of predict ALS progression. And it may be that one day we can predict progression for a given person, treat them with a drug, and see where they end up relative to that prediction. Maybe we don't need a placebo. But we need very good predictions if we're going to do that, and there's a lot of work to be done to get there. So I don't want to give people the impression that this is here and we're just not doing it. We have a lot of work to do to see if we can get to that point. That's one strategy that we can use. Okay? And so I think that's a real, and Jonathan's been involved in trying to refine these algorithms. But there is I need to underline this very clearly. There is still a lot of work to be done to get to individual level predictions. We're getting pretty good at binning people, faster than average, slower than average, if we look in certain data sets retrospectively. But we're trying to bring this sort of forward into fruition. And I'll just end on this note. We have a number of trials. I didn't go through all of the ongoing trials. I think that's a long list. We're lucky that is a long and growing list. People who are involved in trials say that this is important for them because it gives them hope. If we look in databases, people who are involved in trials tend to have slower disease progression, not because they're in the active treatment or the placebo arm, but for some reason that we're not 100% sure about. And if you're somebody who wants to be involved in trials, it can be a dizzying array of options out there. You obviously want to talk with your clinicians. You can also visit the Northeast ALS Consortium webpage at neils.org. And there are ways to search, I've sort of circled them here, but there are ways to search for ongoing trials and even search for sites that are enrolling for those trials. And I would just encourage you to sort of, think about whether that's something you'd want to do. Thank you.